Good evening, everyone. My name is Arabella Duffield, and I am Chair of the Weizmann UK. Thank you for joining us online for our annual From Research to Reality lecture. Of course, I'd much rather be seeing you in person, but for now we'll have to make do with this virtual world. This year really has been an extraordinary moment for those working at the cutting edge of scientific research. And in a moment, I will hand you over to Professor Roee Ozeri from the Weizmann Institute, who is the Vice President of Resource Development. He will give us an update on the impressive work the Institute has been doing on COVID-19. And given that it seems that this disease and potentially other global pandemics will be with us for some time to come, he will share some new plans from the Institute on where we go from here with infectious disease research. But before I put you in his capable hands, I would just like to say a few brief words about our headline speaker this evening. Professor Ruth Arnon really is that rare thing, a living legend. Speaking as someone who's had a lifelong interest in science and a long connection to the Weizmann, it is impossible not to be aware and even a little bit in awe of Professor Arnon. She is a shining example of what can be achieved by women in science. And if every young scientist embarking on their career today were aspiring to achieve even a half of what she has achieved, they will be aiming very high indeed. She is perhaps best known for her role, along with another Weizmann legend, Professor Michael Seller, in developing the blockbuster drug Copaxone, which has helped countless people with multiple sclerosis. She has also made significant contributions to the field of cancer research. It is possibly a lesser known fact that Ruth Arnon and her husband Uriel are also philanthropists. They have given transformational gifts to establish and equip the Ruth and Uriel Arnon Scientific Education Campus at the Institute, which aims to nurture excellence in the next generation of Israel scientists. And as if all that was not enough, she is on the brink of the very final stages of making a universal flu vaccine a reality. And it's this latest achievement that she's going to talk to us about this evening. It really is the most enormous privilege for us to have her with us, and I won't keep you from either of our guest speakers for a moment longer, except just to say there will, of course, be a chance to answer questions at the end. Roee will be our moderator, and if you'd like to ask a question, you can either do so via the comments function, or you can email them to samantha@weizmann.org.uk. Thank you, and I hope you enjoy this evening.
Thank you very much, Arabella. And thank you everyone for joining us this evening. My name is Roee Ozeri. I'm the Vice President of Resource Development at the Weizmann Institute, and on my spare time, I'm a quantum physicist. I build quantum computers. But since December, after spending 25 years in physics labs, I was asked to step in and serve as the new VP of Resource Development as part of new management that came into office in December, headed by our president, Alon Chen, who is a neurobiologist. I know that most of you are familiar with the Weizmann Institute. For those of you who are less familiar, we're a research institute. Compared to other research institutions in the world, we're not very big. About 250 research groups. We're not a university. We don't have a bachelor's degree. We do have a graduate school. And our size and the fact that we don't have very heavy teaching programs turned the Weizmann into a very interdisciplinary and collaborative in spirit and ideology, if you like, research institute. And that means we hold research programs in all life, exact, and natural sciences. We don't focus ourselves on one particular scientific aspect or another. In fact, we don't have a scientific agenda at all. What we believe in are people. We try to bring in the most talented, creative people we can recruit and we let them follow their dreams without trying to steer them in one way or another. Having said that, as new management, we had to survey the needs on campus and then come up with programs that would cater these needs and build flagship programs. These are strategic research programs that look at the needs on campus and bring the resources that are needed, the tools that are needed in order to fulfill these plans. And the three flagships we identified and embarked on are the following. The first is a big initiative, a big institute in brain and neural sciences. Here we're capitalizing on the fact that Weizmann is an extremely interdisciplinary place, and we're moving forward, pushing for the next revolution in life sciences to occur in brain research. Brain research is highly interdisciplinary in nature, combining life sciences with psychology, with computer science, with physics and chemistry, and many other fields of research. A second flagship surrounds around the research of integrating artificial intelligence into all our scientific endeavors. This is really an initiative that is meant to deal with the immense overload of data that scientists today need to deal with. And the third flagship is a flagship in astrophysics and particle physics, in which we're actually launching the first Weizmann space program. So in fact, it's the first space program that an Israeli academic institution is launching, and we actually have a satellite that we plan to send to space in 2024. So as I said, as new management, we assumed our office in December 2019, very excited about our strategic plans, and then within 3 months, the world has completely changed. The way the Weizmann Institute work changed, the way everybody worked has completely transformed. And of course, we needed to react to that. Presence on campus was limited. We had to maintain social distancing like everyone else. On the other hand, despite the fact that previously there was very little research of contagious disease at the Weizmann Institute, within 3 to 4 weeks, we identified over 65 research initiatives towards trying to come up with an answer for this horrible pandemic. As management, we understood that we needed to support that, and that's why we launched a coronavirus response fund, for which I know that many of you have contributed, and I'd like to thank you from the bottom of my heart for stepping up to this challenge at these pressing times. These initiatives really covered multiple directions, from hardcore research into developing new drugs and vaccines, all the way to coming up with smart algorithms that allow to identify breaking foci of the disease well before they're identified by RNA tests, through improving the efficiency and the volume of RNA tests, as well as antibody testing, all the way up to finding ways, strategies, and models that will help with exit strategies from lockdown. For a short, brief period of time at the beginning of the pandemic, we actually turned some of our research laboratories into a facility that would support the Israeli national effort in running RNA tests on the population. And I think that the fact that we could see so many initiatives on campus is really a testimony, if you like, to the fact that we don't have very strong scientific agendas, and that allows our scientists to be relatively agile in the way they can respond to changing realities. At this point, we understand that this episode of the pandemic was not met. Humanity did not meet this pandemic with sufficient readiness. We understand that looking forward, we need to make sure that we're ready for the next pandemic if it comes. And that's why we're also inaugurating an Institute for Infectious Disease that would make sure that infectious disease research stays with us and the lessons are learned. And I think this is a great time to introduce one of our most prominent immunologists. In fact, she's one of the most prominent immunologists in Israel, if not worldwide, Professor Ruth Arnon. Professor Ruth Arnon joined the Weizmann Institute in 1960, and since then I think she's probably been in almost every leadership role at the Weizmann Institute, from a dean of the biology faculty all the way to vice president. Professor Arnon also served in many leadership roles in Israel and around the world. She was the president of the Israeli Academy of Sciences and Humanities. She was the president of the European Federation of Immunological Societies, and she was the president of the Association of Academics of Sciences in Asia. Professor Arnon won many, many prizes, and I'm not going to list them all. I'll just mention a few. She won the Légion d'Honneur in France, the Wolf Prize, the Rothschild Prize, and the Israel Prize. And Professor Arnon is not just a leading academic. She also had huge impact on the pharma industry. She's one of the co-developers of Copaxone, which is a main drug which is helping patients of multiple sclerosis. And today Ruth will tell us about her new adventure and hopefully her new huge impact on the pharma industry, which is a universal flu vaccine. Please, Ruth, the stage is yours.
Thank you. First of all, I want to say how pleased I am to be with all of you this evening. And I would like to thank Arabella and Roee for their kind introduction. I'd like to mention in the beginning that I am a great believer in the fact that in universities and research institutes like the Weizmann Institute, the main emphasis should be on basic research. And I define basic research as that the purpose of which is to increase the amount of knowledge and the use of this knowledge. But sometimes when we do basic research, we step on something that could be applied. It never can be applied in a university or research institute. For it to be applied, the technology has to be transferred from the research institute or the university to industry, because only in industry you can develop the product. And in the case of Copaxone, for example, we did only basic research. We did not even intend to develop a drug. We didn't think in this direction. We wanted to study the mechanism of multiple sclerosis. And in order to that, we studied an animal model in which the disease, a similar disease to MS, is induced in the animal by injecting a brain protein. So we synthesized a polymer that would be similar to the brain protein. Actually, we wanted to induce the disease with it because we thought that this will give us a very good tool to study the mechanism of the disease. But what happened is that this polymer did not induce a disease at all. On the contrary, it could compete with the protein and inhibit the disease induced by the protein. So once we saw this, we saw the potential of it as being a drug. So we took out a patent that was called a therapeutic copolymer. Once we had the patent, we transmitted, or we transferred the technology to the Teva company, because only at a company, at a pharmaceutical company, this could have been developed as a drug. So I mentioned it as a general point. Now, the same thing actually is in the case of influenza. I'm an immunologist and would like to, I'm studying the phenomena that are related to the immune system. And we know that the immune system protects us against many infectious diseases by the use of vaccines. In vaccines usually are based on the agent that is causing the disease, either bacteria or viruses. And what is used for vaccination is the agent either killed or attenuated, or in some cases, an isolated protein from this agent. This is particularly in the case of bacteria. Now, we asked ourselves, okay, if you can immunize with a protein, do you need the entire protein for inducing an immune response? And we studied a very simple protein, an enzyme that is called lysozyme. And we saw that in order to induce an immune response against this protein, we don't need the entire protein. We can take a small segment that is called peptide of this protein, use this segment for immunization, and induce an immune response that will recognize the entire protein. So we asked ourselves, okay, if this is good for a simple protein that we study, will it be good also for a protein that is part of a virus or any other disease-causing agent? And in the beginning, we used, just because it is much simpler to study in the laboratory, we used a virus that is not infective in men or not even in animals, but is infective to bacteria. It kills bacteria. This virus is called bacteriophage because it kills bacteria. This bacteriophage has an envelope, and this envelope contains a particular protein that is called coat protein. We identified a certain segment or peptide of this coat protein of the bacteriophage. We prepared it synthetically, attached it to a carrier, immunized animals with it, and studied the antibodies that this peptide induced in the animals. What we saw was that these antibodies had the capacity to inactivate or kill, actually, the bacteriophage. So that after the bacteriophage was subjected to these antibodies, it was not active anymore and it could not kill the bacteria. Now, nobody is interested in being vaccinated against a virus that kills bacteria. So we wanted to see if the same principle will hold also for a virus that is more interesting to us, a virus that is causing a disease in people. And we chose influenza, not at all because we wanted to prepare a commercial vaccine against influenza, but influenza is a virus that has been studied for many years, and a lot is known about the various proteins that are components of this virus, which protein is important for protection against it, or for infection, which protein is not important. So it was easier for us and more convenient for us to look at influenza as a model system for looking at the possibility of not using the entire virus or entire protein of the virus for inducing a response immune against it, but using a peptide, a segment of a protein, in order to induce an immune response. So we looked at the virus, and the first stage in our research was selecting one protein that is called hemagglutinin, or the H. You know that influenza is defined by H and N. These are two different proteins. So we took the H protein. We identified one segment of the protein that we thought that is important for inducing an immune response. And we immunized mice with this segment, with this peptide. Now, mice are sensitive to the same strains of influenza that are infectious in men. So after immunization of the mice with this peptide, we subjected them, we exposed them to the virus, and we saw that in in contrast to mice that were not immunized, just normal mice, that after being exposed to the virus, most of them, 90% of them, contracted the infection, and if the exposure was high enough, even died of it. 90%, or I don't remember if with this particular peptide, a significant percent of the mice did not contract the disease at all, and remained completely healthy. So this was by using just one peptide. Then we thought maybe we could increase the effect by, and we added 2 additional peptides from two other proteins, and we used the combination of these 3 peptides, and we saw that when we immunized the mice with the combination of these 3 peptides, we could protect them completely from infection by exposure after being exposed to the influenza virus. So wonderful. We had a very nice vaccine for mice against influenza. So how do we continue from that in order to develop something that is more relevant to men? So first of all, we used a system that is called of humanized mice. What are humanized mice? These are mice in which by irradiation, you kill completely their immune system, and afterwards, you transplant them with human blood cells, white blood cells. So they remain mice, of course, but their immune system is the human immune system. When we had this, we did not develop the system. Actually, this was developed by Yair Eisner at our laboratory, but we used mice the technique that he developed. And we then identified some peptides in the influenza virus that we thought that, from various considerations that I will not go into, that we thought that will immunize or affect the human immune system. And we used the combination of 4 such peptides, and we immunized the humanized mice. And we thought that with the combination of these 4 peptides, the mice that we vaccinated were completely protected against infection with influenza. The control group, all of them died. In the vaccinated group, none of the vaccinated mice died, or 90%. We got between 90 and 100% protection against infection. Not only that, even several months after the immunization, the vaccination, the mice continued to be protected against infection. At that point, I decided that I have finished the basic research that can be done at the Weizmann Institute. We published a patent. The patent was published by YEDA, and I moved to another topic, to another project. Soon after that, a group of 3 young entrepreneurs, they visited YEDA, and they asked YEDA if they wanted to establish a startup company, and they looked for an idea. And YEDA proposed to them several possibilities. One of the patents that they have studied, that they have looked at, is our patent on these 4 peptides that induced protection in humanized mice against influenza. And they decided that they want to take it on and to apply it as a commercial project for the development of a vaccine against influenza. I forgot to mention the major point. The peptides that we selected were peptides because influenza virus is different from all other viruses, or from most other viruses, in the sense that every year they undergo changes in their 2 major proteins, H and N, and therefore the vaccine that was prepared against the strain of last year will not be protective against the strain of this year, and the vaccine that is prepared against the strain of this year will not protect against the virus that will be the circulating strain next year. Now, when we selected the peptides for vaccinating, we selected peptides from regions of the protein viruses, of the virus proteins, regions that do not change between different strains. So we looked for constant regions for two reasons. First of all, we thought that if we have a vaccine, it will be effective against several strains of the virus, or many strains of the virus. And so this, of course, is the first reason. And the second one is that in this case, if we can produce a vaccine, it will be a kind of a universal vaccine. So the peptide that we selected, after immunizing with them with our synthetic peptides, we exposed the mice to different strains of influenza, and we saw that indeed our combination of peptides protected the mice against every single strain that we tested. I don't know if, I cannot say that it protects against all strains, but against all strains that we tested, and we tested quite a few from different classes, H and B and so on, influenza A and influenza B. And so we think that it will be protective against many strains. So as I said, when we got these results, we published the patent. I finished my role as a basic research person. And at that point, these three entrepreneurs came into the picture. They signed with YEDA an agreement of that, according to which they can develop it into a product. And they established a company that is called BiondVax. And this company continue to the research. They realized that using combination of 4 different compounds is not a good idea for developing a product. So they, after doing some more research, they have come up with a single string of it's a very long peptide, very long polymer, as a matter of fact, that is composed of 9 different, the 4 that we had, and 5 additional peptides of influenza proteins. And using this synthetic polymer, they vaccinated mice, humanized mice, and also transgenic mice that resemble more the human antigenic and immunological properties. And they saw that their protein, their synthetic protein, is capable of protecting, leading practically 100% protection in mice, between 95 and 100% protection. Following that, they started to do some clinical trials. First of all, it's phase 1. Phase 1 tests only for safety, and they have shown that their synthetic protein is completely safe and does not cause any side effect, and any adverse side effect. And then they did 6 different phase 2 trials, namely immunizing, vaccinating people and checking their immune response, antibody response, and mainly cellular immune response that is supposed to take part in the protective effect. Of course, they did not subject the individual, like we do in mice, to the infective virus, but they looked only for the immune response in the vaccinated individuals. And as I said, they performed 6 different phase 2 trials, each of them in several hundred people, in different age groups. One of them was in the elderly, since the elderly are supposed to be more sensitive to influenza. So this particular trial included individuals between over 65 years old. The oldest one was 91 or 94, I don't remember. Anyway, in all of them, they saw good immune response, although they do not have any information about the protection against infection. The next stage is a phase 3 trial. The phase 3 trial was something that they did in coordination with EMA. EMA is the European Medical Agency, which is the parallel to the FDA in the United States. And in coordination with them, they did a phase 3 trial over 2 years, last year, the summer of 18-19 and the summer of the winter of 18-19 and the winter of 19-20. And altogether, more than 12,000 people participated in this trial, all of them at the age of 50 and over. Half of them received placebo and half of them received the vaccine. And next week, we are supposed to receive the results of this trial, and we hope very much that they will show efficacy of this vaccine. If indeed it will show, we will observe efficacy. What we have shown is that the vaccine in mice is effective against every single strain of influenza that we have tested. By the way, the EMA required that the trial will be done over 2 years to show that indeed the vaccine is effective against 2 strains of influenza that are the circulating strains in these 2 different years. So we are now, as I said, in the last stage of this I would say, trial to obtain a universal flu vaccine, and in a short time, we will know whether it has been successful or not. Thank you very much for your attention.
Thank you very much, Ruth, for this enlightening talk. It's amazing to see how progress is actually moving from the basic research laboratories all the way to the clinic. We will, before we move into the Q&A session, we will summarize this with a short movie on vaccines, and after the movie, we'll go into the Q&A session. So if you'd like to ask Ruth questions, please email them to Samantha.
A virus is not really a living organism. It's just genetic material, either DNA or RNA, surrounded by a protein coat that can replicate, and this is what we call a virus. All viruses have a receptor on their envelope, and they can recognize their host cell and get into it, and then by different mechanisms, they can make the host cell replicate the genome, generate viral proteins, and basically make the cell a viral factory that will now generate hundreds or thousands of new viruses that will bud out the cells and will infect new cells. So this is the common thing for all viruses.
We know about the immune system, even about becoming immune, like vaccine, for thousands of years. The Greeks found out that if one is exposed to a disease, then it becomes protected during second exposure. But then nothing was done about it for thousands of years. And then came the vaccination, which was the first immune manipulation that provide protection.
Jenner was a country doctor, and he noticed that cowgirls had beautiful face because they suffered less from smallpox than others. And he connected the cowpox and their exposure to cowpox to their being protected against smallpox.
Edward Jenner did inject a dead cowpox virus to a kid, and because the antigen was very similar between the cowpox and the smallpox, it made the child resistant to now smallpox.
Vaccination was one of the most important discoveries in the field of immunology. Vaccination saved lives of millions, billions.
The contribution of vaccines to public health, this is the most important and cost-effective until today for all diseases.
You may not know this, but vaccine comes from the word vaccine, which is in Latin means cow.
And vaccinia virus is the virus of the cowpox.
For us to advance beyond the state of the art, where the field is, we need to take risk. And you cannot take risks if there is no philanthropists or people who come and strongly support unexpected science.
And this is our hope for the future.
And we have now new tools, new generation tools of sequencing, of producing antibodies. And by combining that, just imagine what could we do?
Okay, welcome back. We already have questions coming in. So one question, Ruth, is the following. You have had a very successful career in immunology, covering an unusually wide range of disease areas. Tell us a bit about what has motivated you to pursue the subject areas that you have.
Could you repeat the question? I did not hear the last part.
The question is, in your career, you really covered many topics in immunology. How did you choose your research topics?
It's very difficult to tell. Most of the time, I was not guided by the final purpose, but it developed naturally. You do some experiments, you have some findings, then you see that you can use this finding in order to go to the next step and the next step, and finally you get into a question that could be relevant to and interesting even from a practical point of view. It's not something that I planned in the beginning.
So it's curiosity-driven research.
Absolutely, curiosity-driven, and I define basic research as the research that the purpose of which is just to increase the amount of knowledge and our understanding of the phenomena in nature. And this was my guide in selecting what I was doing. But somehow it led me into questions that are of interest even from a practical point of view.
That's a good advice to give young scientists, I think. So another question is, clearly there's a lot of interest in the results of the trial. Can you let us know when the results will be published or where they will be published? How can we know?
First of all, this is a problem that will be held by the company. The trial has been performed by the company, by BiondVax, and they will handle it. I guess that once they get the results of the trial and the discussion with EMA about it, they will publicize it, I am sure, as soon as they can.
All right. So the next question, Ruth, is clearly there's a global race to reach these vaccines. So are there other attempts to reach a universal flu vaccine? And if they are these attempts following the same procedures and ideas of your vaccine, or these are different approaches?
There are other groups that are trying to develop also a universal flu vaccine based on other assumptions and not on peptides. I don't know of anybody else that is using a synthetic compound. They are using either one of the proteins of the virus that is less changing, or regions of the proteins that are changing less, or using monoclonal antibodies that are known to neutralize the virus. So there are various approaches that are being taken, but none is equivalent to the approach that BiondVax has taken.
So this is a unique solution in a way.
Yeah, this is a unique approach, I would say.
So another question, and that's an interesting question today, with a COVID-19 health crisis, Is there an increased urgency in actually acquiring a universal flu vaccine? Winter is approaching rapidly, so do you think that there is an increased urgency in reaching this universal flu vaccine because of COVID?
For the COVID? Potentially, I think yes. And I'll tell you why. Because coronaviruses are very common, and usually they cause common cold that is not a very serious disease. However, we know at least of 2 other coronaviruses, the SARS and the MERS, that have been almost pandemic, and now the COVID-19. So I think that there is a point in looking at the proteins of the sequence of the proteins of coronavirus and looking for what is common among those 3 strains, and maybe using, if we see that the influenza, the universal vaccine works, using a similar approach, namely focusing on regions or peptides of the virus proteins that occur in pandemic strains and do not occur in the non-damaging simple corona that causes a common cold, and focus on them, and maybe using a similar approach, try to prepare a vaccine that will be effective not only against COVID-19, but also against another pandemic strain that may come in 10 years from now.
So Ruth, another question is, from the time of FDA approval, how long would you anticipate that it'll take before we can find it on the shelves?
BiondVax has built a plant that they can produce several tens of millions of doses per year. This is definitely enough for Israel, particularly since if this vaccine works, then you don't have to be vaccinated every year. Every 5 years, like against the tetanus, will be enough, maybe even less than that. So it certainly will be enough for the population of Israel, but not for the population of the world. But they are already in contact with some plants that if everything goes well, they will be able to use the same procedure for preparation of a large amount that will be available.
So actually, you described very nicely the full trajectory from the laboratory all the way to stage 3 clinical trials. How long did it take? How long did it take from the first ideas to the point that we are now?
It took BiondVax more than 10 years of clinical trials. First, the phase 1. First of all, the building of the commercial process for vaccine for preparation, and not exactly what we have prepared. It took Teva, by the way, with Copaxone, it took several years, I think 9 years, until from the time that we finished the clinical trial and Teva went, started or signed the agreement with the Weizmann Institute in 1987, and they came up with the Copaxone for treatment at 1996. So it takes time. Here it will take less because everything, I think, is simpler, and BiondVax could come up with it within several months, but to produce it in a amount that will be sufficient for on a global level, it will take a longer time.
Okay, one other question reflects the concern of people with vaccinations and the long-term effects. So clinical trials, they're testing the effect in over the course of 1 or 2 years, as you described. Is there a worry about longer-term effects that are not being tested in clinical trials?
I have no information about that.
Okay. One question that came up is, how big does a peptide have to be in order to obtain an immune response?
From our studies, we know that a peptide that is between 15 amino acid residues and 25 is, from my point of view, is the ideal size for a protein to have an antigenic imprint and to be effective. Sometimes, if it is smaller peptide, it has to be attached to a large molecular weight carrier. But if it's if like in the case of BiondVax, they attach the peptides to each other, actually they have a construct that is built from all the peptides, and you don't need a carrier.
So another question takes us back from the more technical parts to the practical parts. What happens if the FDA says no after a third stage clinical trial? Is that the end of the road, or can you go back and try a variation to this approach?
This will have to be decided by BiondVax. I don't know.
Okay, I think there's one more question here that says whether, I think you mentioned 5 years, is there any likelihood that the universal vaccine would actually be lifelong vaccination? Why 5, and if 5 years or 10 years, why that?
There is no reason to say 5 years. The reason that I said is because it is known for tetanus vaccine. I take tetanus as a model. So if you are vaccinated against tetanus, the assumption is that it will be effective for 5 years. And if you have an injury and you have been vaccinated more than 5 years ago, you are required to take another vaccine. So I assume that this is just a number. It could be 5 years, it could be 7 years. Nobody knows for sure. I don't think that it will last for a lifetime, because every vaccination needs a boost from time to time.
Okay, you know, as an immunologist, we mentioned before that the world was largely unprepared for the COVID-19 pandemic, even though we have seen bursts of epidemics, in particular in the Far East in the last 20 years. What do you think is a good lesson for us to learn as a scientific community and, you know, as countries to be better prepared for the next time? What are the things that we need to do?
Well, you can never predict whether there will be a particular virus that will be very harmful and very infective like the COVID-19. I think that in the case of the COVID-19 that belongs to the SARS family, had the world been prepared in advance and prepared enough vaccines against MERS and SARS, or something that can recognize the two of them with a chance of recognizing another strain, I think that this could have been of help. But I don't know. Both the SARS and the MERS were relatively short and not long. They were not as had the same effect as the COVID-19. So nobody took them seriously enough in order to prepare enough of the vaccine that will be ready for the entire world to be vaccinated in case something else of this sort comes along. I think that now, after the COVID-19, I think that there will be more thought about in this direction.
So Ruthie, here's a question I think you'd like. What do you consider as your greatest achievement as a scientist, and why?
The greatest achievements of scientists at all?
No, of you, your greatest achievement as a scientist.
Mine? I think that if the flu vaccine will indeed be a universal flu vaccine that will really protect us against the flu, against a potential flu pandemic that we did not have until now, not recently, not for 100 years, but if it will be protective against it, I think that it will be, although Copaxone I think that it does great for MS patients, but I think that the number of people that will be helped by a universal flu vaccine will be much, much larger than the number that is being helped by Copaxone.
Okay, so another question here is, if you could give one piece of advice to a young scientist just starting her career in the scientific world, what would it be?
What I would say is, think of something that excites you most and that you really love most, and decide to go in this direction. Even now, in my advanced age, when I wake up in the morning, the thing that I love most to do is to either to go to the office or to be engaged. These days I do it at home, to be engaged in scientific issues that I'm involved in. So, and I think that this is the most important thing. One has to love what they want to do, and if you love it, you'll do it well.
Okay, I think this was great advice. I hope there's some young scientists that are hearing us. So I think at this point it's a good opportunity to thank you again very much, Ruth, for your enlightening and fascinating talk and lecture, and for the interesting answers to all the questions. And I'm now transferring the stage to Sheridan.
Thank you, Roee. And I think right now for all of us, it's really great that we've had some good and positive news, and really, Ruthie, I think all of us watching tonight are eagerly awaiting the outcome of those FDA results. So for those of you watching who don't know who I am, my name's Sheridan Gould, and I am the Executive Director of Weizmann UK, and I have the best job this evening. Actually, my job is really to thank the speakers and to thank you. I think this has been a really uplifting and fascinating talk that we've heard, both from Roee on what's going on at the Weizmann Institute right now, and certainly from Ruth on her outstanding career as a scientist, and especially this latest work that she shared with us this evening. I mean, it's really clear with everything going on in the world right now that the introduction of a universal flu vaccine would be of absolute benefit to humanity. And actually, it's the core value of what the Weizmann Institute's all about. And I really couldn't be prouder to be associated with this remarkable institute and its scientists that are all working at the forefront of what is possible to achieve. And Ruthie, you really exemplify that for us this evening. So really, my job is simple. As I said, I'm really here to say thank you, and I will repeat my thank you again. I'd like to thank Arabella for taking on the role of chairing Weizmann UK. We're really so happy to have you. Your family's been involved with the Institute for so long, and it's so lovely. The third generation of this family, you know, taking up that leadership role is truly valued. Roee, as I said, the role that you and your management are doing at this time of crisis, we're thrilled to have you in that role, and we can't wait to invite you to London so we can meet you here in person. And of course, finally, the biggest thanks of all goes to Ruth for the remarkable career she's had. I know most of her career has been spent at the Weizmann Institute, and as Arabella said at the beginning, not only are you a great scientist, but the Institute really enjoys the benefit of your and your beloved Rooe's fantastic philanthropy that you've generously given to the next generation of scientists. So really, I'd like to thank all of you for watching and for sending in these fascinating questions. I have to thank the production company, Neil Oakshott and his team at Eclipse, for making this all possible. And I of course have to thank my team at Weizmann UK for what they've done. I can't really impress strongly enough how important at the moment in this current pandemic that staying together in this virtual world and being part of this community. It's really important to us that you have actually signed up and tuned in tonight, and we're really, really grateful. We have recorded the evening, so a recording will be available on our website from next week, and I would urge you to, if you can, share it with friends and family. We really would like as many people as possible to be aware of the work that Ruth has done. I should also say in my role that we have obviously invited you tonight to join free of charge, which is absolutely fine. But if you do feel that you're able to make a contribution to the amazing work of our scientists, we have got a donate button on our website, and I would encourage you, if you're able to make a contribution, it would be greatly appreciated. So really, I think that wraps it all up for this evening. Ruthie, we are all eagerly awaiting the news of those.
Oh my.
Excellent. And we will share it with everyone when it comes through. And again, so really all it is for me to say at this point of the evening is stay safe, stay tuned, and thank you. Good night.
Thank you.
I enjoyed being with you tonight.
Thank you
very much.